Alright folks, so we've gone over evidence for evolution, the process of evolution through natural selection, and how we classify the life that we see on Earth. Again, remember, nature doesn't care, but we do. We need to put things in little boxes so that it's easier for us to understand. Now, this topic is how we decide which box to put things into. So here's this diagram showing the three domains of life as we know it. We have bacteria, archaea, eukarya, and the way they decide how to divide each of these into different categories is by looking at different characteristics, whether it's morphology, anatomy, cytology, that's the cell structure, phytochemistry, secondary metabolites, such as, okay, you have the metabolites that you need in order to stay alive. Things such as ATPase or other enzymes that help with the primary functions, breathing, oxygen transfer. Well, that's mostly diffusion, but through the processes of, if you're a plant, photosynthesis, enzymes responsible for cell respiration, those are all primary metabolites. Secondary metabolites are chemicals that are not necessary for life but make life easier, such as poisons, toxins, and other chemical substances that can help you win a fight against a predator, help you overcome prey. Those are all secondary metabolites, and different groups have different secondary metabolites. So if you categorize those similarities, that's phytochemistry. You can also look at chromosome numbers, which we've talked about before, and also the one that has the finest resolution to date currently. This might change in the future, change in technology, we get new data that we can look at, but currently the most information that we can get is through molecular differences, the DNA and protein. Now, IB usually puts this in the previous topic, but I want to put it here instead because it helps us visualize how we categorize things. So first, the basic structure. You have dendrograms, which are branching trees that let you know similarities, let you visualize similarities between different groups. You have your cladograms, which are your basic branching trees, and then you have the phylograms, which are a modified version of this. And if you take a look at it, notice that all the branches are pretty much the same length for the cladograms, but phylograms, you have different branch lengths. The cladograms, tree 1 and tree 2, they say this exactly the same thing. They're just whether you have square brackets or whether it goes on a diagonal. Now, for phylograms, you can have either square brackets or diagonal, but usually you do it by the square brackets because the thing about phylogram is it shows time, by how long that branch is. So here, taxon A, B, and C have the same relationship. B and C are closer related than they are to each other than they are to A. All right, so I messed up, so you're going to get a little bit shift of view. But, okay, again, A, B, and C, B, C closer related to A. They had shared some common ancestor somewhere back there, and then that common ancestor split off to taxon A and the ancestor to common taxons B and C. And not soon thereafter, that ancestor split into B and C, and they have been extant for quite some time. Whereas in this one, same case, A, B, and C, B, C closer related, way far back in time, ancestor A, B, and C existed, but the ancestor to A emerged and the ancestor to B and C emerged and stayed around for a long time, and it's only fairly recently that B and C have split off from whatever ancestor that they shared that was not the same ancestor that they shared with taxon A. So if we go back to the three domains, you can see here bacteria, eukarya, and archaea all shared a common ancestor, but way back in time, they split off. Bacteria had a long development time, and then now they're starting to, well, not now, but then they diverged off. But you can see by this branch length versus the branch length between archaea and eukarya that archaea and eukarya are actually closer related than bacteria, and we have a lot of data that supports this particular branch length on the tree. So this one would be a phylogram, not a cladogram. Cladistics is a science of putting together those trees, and they use various traits and various data to help them figure out the relationship between the different species. So all of them, cladistics mainly is looking for shared ancestry. And by looking at the ancestry, you have different traits that different taxa share, such as plesiomorphic traits. These are more ancestral traits. So if you look at this picture, ancestral trait here would be their vertebrates. And these would be plesiomorphic traits. That's something that's more ancestral, developed earlier, and all the taxa share it. Then you have the apomorphic traits. These are the traits that develop later, such as for the entire group, everything above vertebrate would be an apomorphic trait. However, if you're looking at fish, apomorphic trait would be forelimbs, amniotic egg, hair, and preorbital fenestra. So an ancestral plesiomorphic trait is an ancestral trait of a group that you're looking at that everyone shares, and apomorphic traits are traits that are further up, that develop later, that help you distinguish between the different taxa. All right. Now you have derived states, which are adaptations of plesiomorphic traits. They come around much later than the plesiomorphic trait, but that plesiomorphic trait, such as, let's take an example, leaves, has been around for a very, very, very long time, but if you're able to modify that leaf and use it for a different function, that would be called a derived state. For example, that leaf, you know, all those flowers, those beautiful flowers, or for example, bougainvillea is a very good example because all those petals of the flowers, they're just adaptations of leaves. So the leaves have been around for a long time. Flowers, not as long. Plants have been around for a long time, but angiosperms arrived much later on the scene, and those flowers came from the leaves. Bougainvillea, if you look at it, those beautiful petals on the bougainvillea, they're not actually flowers in themselves. So bougainvillea, I'm not sure of this, I haven't researched ancestry, but I would, from looking at that, call that a transition state from actual leaves to full-on flowers. All right, then you also have all these different traits, and if you gather them all together, look at them, you can try to use that data in a parsimonious manner, which is basically looking at what's the shortest path from A to B to C and all these collections of data to figure out where everybody fits in relation to each other. And if you have a set of taxa that fall together and share a common trait, such as if you're looking at mammals and they all have hair, that would be called a monophyletic clade. If you're looking at the groups of primates and the groups of rodents, they both have hair. So in that case, if you're looking at that level, they would no longer form a monophyletic clade because two different sets, the primates and the rodents, have that trait. So that would no longer be considered monophyletic. So while cladistics is looking at shared ancestry, phylogeny is looking at things in a slightly different view. It's still looking at ancestry, but through the lens of evolutionary relationships. How are the different taxa related to each other by how they evolved? And the way you look at it is through conserved genes a lot of times, and you have mutations that occur. We all know that in the DNA, but those mutations occur at certain rates, and different mutations will collect, and you'll compare the genetic sequence of the different taxa to see, okay, does this one have that mutation or not? And you can start using those as characteristics to do cladistics and see what ancestors are shared. You can also look at analogous and homologous features. I'm not going to go into that because we covered that already. And that can also be applied to molecular data. Have they achieved a certain ability through the DNA? Did they achieve that ability through ancestral evolutionary lines, or did they all converge? Did they convergent evolution onto that particular trait? All right, and you don't just use molecular data. Even before we had molecular data, they have been doing phylogeny. A lot of times it was with physical characteristics. So you don't throw away the old, you keep the old, but then you just add more data, and that will refine and support your conclusions. So some of the data that can help support the creation of these phylogenetic relationships would be the DNA and RNA sequences, so just the nucleotide sequence, A, G, C's, and T's, which one switch, and remember we have over 6 billion of those in our bodies, or each of our cells actually, and then if the DNA sequence actually codes for something, you can look at the amino acid sequence. So DNA, if you remember, is degenerate. The code is degenerate, meaning you can have multiple codons code for the same amino acid. But if you have different amino acid sequence, that can change the entire structure and function of that protein. If another example that we've covered is in sickle cell anemia. The sickle cells, you only change one DNA, you have a single nucleotide polymorphism, and that changes that particular amino acid, which changes the entire. We have to remember, though, that a phylogenetic sequence or a phylogenetic tree also looks at time, not just relationships. So if this one up here, this picture I have up here is actually a cladogram, not a phylogram. Phylogram, remember, will also show time of divergence. So one way we try to figure out how long ago differences occurred, especially if we're looking at DNA and RNA sequences, is by the use of what's called an evolutionary clock. Now, these evolutionary clocks are actually estimates. They're not set in stone. They're calculated by researchers who look at different taxa, different species, and look at the changes that they see over thousands to 10,000s of years, and use that to try to figure out, okay, on average, how many changes in the DNA sequence do you see over so many thousands of years? So usually, a lot of times, depending on the time series that you're looking at, you look at either more stable or less stable coding sequences, conserved sequences, or just sequences that are just in between the coding sequences, because those areas can evolve much faster. But remember, these are hypotheses. They are estimates only. One that they use a lot is mitochondrial DNA, because it is highly conserved, has to perform a function. Again, back to the sickle cell anemia. If you change the sequence, you have the potential of changing the protein. Change the protein, you can change its function. And that can have potentially catastrophic results. I want to reiterate that all these relationships that I've been talking about, that we currently think is what's true, they're all hypotheses. As mentioned before, they're all ideas, and that we think of how the world works, but as we get more data, that can change, and we have to refine and maybe modify what we knew. Here is an example you see here of the history of the kingdom system. Remember, we have 3 domains: domain Eubacteria, domain Archaea, and domain Eukarya. And within those, we have different kingdoms. At first, in around 1753, we only knew of 2 kingdoms: Animalia and Plantae, animals and plants. But then they looked and they saw, hey, there are single-celled organisms. So in 1866, they added a third kingdom, Protista. Then in 1938, they figured out, oh wait, some of these single-celled organisms are very different from everything else. That got you Monera. And then 1959, they took a look at a fungus and go, wait, these are neither plants or animals, and they are multicellular, so they don't fall in protists, which is basically the single-cell junk category of, hey, if it doesn't fit in anything else, we throw it in here. It's also not Monera, so they created fungi. And then in 1977, they took a look at Monera, the single-celled prokaryotic organisms. They noticed, wait a minute, these 2 groups, they don't have internal membrane-bound organelles. So yes, that's what categorized Monera in the first place. But they do have differences between each other, such as Archaea actually have DNA-associated proteins and several other characteristics. So they split Monera up into Archaea and bacteria. So as we get more data, as we get more information, we might have to update these kingdoms, update our knowledge of all the taxonomy that we have as we go along. That is how science works. We start off with an idea, we keep with it until we find something else, then we either throw that one out or just modify it to fit with what we know now. So things change, and as with technology, we can get more data. As we get more data, we can get a better idea of the relationships of life on this planet.